Are you tired of only learning theory in class when what you really need are practical lab skills that employers are actively searching for in new hires? Over the next 8.5 minutes, I will break down the absolute latest molecular biology techniques crucial for every student hoping to excel in 2025, focusing on methods that are widely adopted and accessible for early learners. Hello everyone, and welcome back to the Labmonk channel, where we bridge the gap between academic knowledge and real-world laboratory competency. For each major advancement we discuss today, I'll clearly explain its fundamental purpose, highlight precisely why it has become indispensable in modern research, and share one actionable step you can take immediately to begin mastering it. 1 PCR upgrades, qPCR-Y, digital PCR, dPCR. PCR is still the backbone of molecular work, but how we quantify DNA changed. Real-time PCR, qPCR, tells you relative amounts. Digital PCR splits a sample into thousands of tiny reactions and counts absolute molecules. That means better precision for low-level detection like rare mutations or viral load tests. If you plan diagnostics or sensitive work, learn both Q. 2. CRISPR advances, prime editing and safer delivery. CRISPR gene editing matured fast. Beyond cutting DNA, prime editing now edits bases more precisely without making big double-strand breaks. This reduces unwanted errors. For students, understanding pGRNAs, nuclease Cas9, and delivery strategies, plasmid, RNP viral, gives you an edge if you're moving into genome editing or cell engineering. 3. Next-gen sequencing. Short and long read. Sequencing went from rare to routine. Short-read NGS still rules for many studies, but long-read sequencing nanopore, PacBio, now reads whole genes or transcripts in one pass. Great for structural variants and full-length isoforms. Portable sequencers let you run experiments outside big cores. Learn sample prep logic and basic data QC. You'll be useful on any sequencing project. 4. Single-cell RNA-seq and spatial transcriptomics. Bulk RNA tells averages. Single-cell RNA-seq reveals cell types inside tissues. Even hotter is spatial transcriptomics. You get gene expression with exact position in the tissue. These methods are transforming developmental biology and clinical work. If you want to work in modern labs, get comfortable with the general pipeline: sample prep, SUBI library, Thousand sequencing, data analysis. 5. Isothermal amplification and CRISPR diagnostics, LAMP, Sherlock, detector for fast, field-friendly tests. Isothermal methods like LAMP amplify DNA at one temperature. No thermocycler needed. CRISPR-based diagnostics, Sherlock, Delaware Tector combine ease and high specificity. These are huge for point-of-care tests and are easy projects for labs focused on diagnostics. 6. Lab automation and low-cost robotics. Automation isn't only for big labs. Affordable liquid handling robots and workflow tools speed up repetitive tasks and reduce human error. Knowing how to write basic protocols for automated pipetters or how to use Opentrons. Style platforms makes you more employable. 7. Imaging advances and super resolution tools. Fluorescent imaging continues to evolve. Super resolution and faster confocal systems let you see nanoscale details in cells. If you're into cell biology, basic fluorescence, staining plus microscopy etiquette, controls, exposure, and quantification is essential. Also, if you found this overview valuable for planning your studies, hitting that like button really helps us determine which deep dive topics to prioritize next for the channel. And of course, please subscribe so you don't miss that follow-up video, and be sure to check the full description for carefully curated links to introductory papers and open source protocols related to every technique mentioned today.